In this lecture titled Methods Overview, I am going to talk about some of the technologies that are driving personalized medicine forward. There are a few topics that I want to hit on in this lecture. First of all, I'm going to talk about some stuff that might be most relevant to industry, and that simply is how these technologies are relevant to drug development, specifically when we might apply our energy to developing a new drug target, versus when we might apply our energy to developing diagnostic tools for deciding who's an appropriate candidate for that drug. Then I'm going to talk about SNP identification, and we'll talk about new SNP identification versus diagnostic tools for identifying SNPs that already exist and are known to be relevant to a particular disease type. I'm going to be talking about specific methods such as dynamic allele, specific hybridization methods, other sequencing tricks. I'll be talking about electrophoretic techniques, and this will be gel electrophoresis versus capillary electrophoresis. I'm not going to talk specifically about the technological differences between the two, but we need to realize that both are in play at different points. But what we're going to see is that I'm going to transition between SNP identification to largely expression profiles. So we'll be looking at DNA microarrays and we'll talk about how these are useful in driving personalized medicine forward. I'm going to be talking about monogenic problems. So let me back up and say that there'll be lots of examples. Once I've talked to you a little bit about the technology, I'm going to move into examples of how these technologies are useful. And in terms of SNP diagnosis, that kind of technology might be relevant to monogenic disorders, in which there are a series of well-defined mutations that are responsible for the pathology. But also we'll be talking about lots of examples of genomics problems in which microarray technology is much more relevant. These other technologies, HPLC, MS, and of course PCR, we're going to push off to other lectures perhaps, and sticking today principally with the methods of SNP diagnosis and DNA microarrays. Let me also, however, say that we'll see some protein arrays too. So keep your eyes open for some protein arrays. So the point is to give you a series of examples that are going to give you a broad but specific picture of the utility of these tools, and less so about the fundamental workings of DNA microarrays, for example. Okay, so when is what technique relevant? So let's think about this y-axis as being the amount of money or time that we put into a particular program or problem, and this is the lifetime of the development of a drug. And so genomics might be a great place to start for the identification of potential targets. So let's say that might be early on, and their pathways. And I'm only going to refer back to EGFR here as an example of how understanding the pathways can be very critical to understanding the efficacy of the drug. Remembering that downstream elements from EGFR turned out to be very important in determining the disposition of these patients. In any case, early on, we spend time identifying our receptors and the pathways in which they're involved. But later, we'll spend our time identifying polymorphisms, and that's where our pharmacogenetics comes in. And of course, we've talked about how drug metabolizing enzymes and transporters are relevant, especially at least to the small molecule set of drugs that we've considered relevant to the disposition of the drug. But of course, there are other examples of how we need to pay attention to polymorphisms in the individual patient and how that affects the drug disposition. After that drug has made it to market, of course, before it makes it to market, but the diagnostic tools become mostly relevant post-market, and so we're going to be looking at therapy-specific genetic tests for use late in the process. Now, What about this cost-benefit analysis? Well, obviously it's not, to call it a cost-benefit analysis is a little bit of a misnomer, especially nowadays that the relevance of pharmacogenetics is obvious to everyone. Instead, it's a matter of when we spend time and energy on what. But let's just look at the numbers just for a second, and these are sort of the back of the envelope, hand-wavy numbers that we're all familiar with. If we spend $200 million on the development of a drug, it might yield us a so-so candidate. Even so, that's going to result in a very large number of prescriptions each year and a very large number of diagnostic tests that are performed each year. And that number only goes higher with the availability of new tests and new technologies. But what I'm trying to get at on this slide is the idea that for a disorder that's well understood, such as cystic fibrosis, we're talking about an enormously large number of tests that are performed annually to screen the population for particular treatments, and this number far outstrips the availability of clinics to perform the work. So I guess there's a couple of ideas here. First of all, of course, we have to diagnose our patients for whether or not they belong in a particular therapeutic category or not, but also just in terms of industry, a shift in focus towards the diagnostic side of the industry is relevant and important and happening. Here's just a nice page for some factoids. Let me point you to genetests.org if you want to see about what some of the tests are. These numbers are very recent. Just last year, this number was 1,200. So just in terms of the clinics available, that number is not exactly constant, and this might be offset perhaps by increases in efficiency, improved technology, etc. In any case, getting back to the idea that the demand far outstrips the availability of testing. Lots of diseases getting tested for, lots of development happening to screen for new diseases or research into new screens. And this list here below, of course, I'm going to circle cystic fibrosis because I'm going to target cystic fibrosis in upcoming slides. This is simply to show you some examples of some of the SNPs that are screened for in these current sets of genetic screens. Let's not spend too much time on definitions here, but it's worthwhile reviewing this stuff. SNPs are clearly single nucleotide polymorphisms, and here's just sort of a laundry list of details. Biallelic sequence alternative polymorphisms are the norm. Tri and tetraallelic SNPs are very rare. Most SNPs are C to T. The frequency is low, and it's even lower in coding sequences, which is pretty interesting given that the coding sequences themselves make up a relatively small proportion of all of the DNA. Most of those are non-synonymous codon changes, or over half. There's a large variation in the number of SNPs across the genome, and I have a slide to show you in just a moment after that. The wrinkle here is in describing what a haplotype is. And a haplotype, simply stated, is just the signature of linked alleles. So what is the allele structure of your genome? That defines your haplotype. And to think about linked alleles, we have to think briefly about linkage disequilibrium. And it's not so fun, and it's an introductory genetics idea, but let's try to review it in a way that makes it relevant to our discussions. So linkage disequilibrium identifies two alleles that segregate in a recombination event. So if we look here at capital A, little b, in which are two parts of on two different chromosomes in the diploid, we undergo a recombination event, and we wind up with this structure. Now big A, little b are on the same chromosome in that haploid cell. They have segregated together in a recombination event. The physically close markers is my shorthand for high linkage disequilibrium. So the linkage equilibrium is going to reflect what kind of distribution of those alleles we might see in sort of normal circumstances. What's the distribution of those or separation of alleles, generally speaking? In the case where those segregate together more frequently than we would otherwise expect, that's a high linkage disequilibrium. They violate that equilibrium. In terms of SNP identification, frequently we'll be looking for an SNP as a haplotype, that is to say, connected with another allele or another marker, so linked. Looking for an SNP, say in the gene that we're interested in, linked to a marker allele that's well described. And I think I can just about leave it at that and say that oftentimes when we're hunting for an SNP, we're not hunting for our SNP per se, but we're hunting for the marker to which it's in strong linkage disequilibrium. Okay, I'm not going to discuss the meaning of this slide. I'm not going to speculate on the meaning of this slide, but just to show you the non-random organization of SNPs in the genome. Here's all 22 chromosomes. We see in red high densities of SNPs and blue lower densities of SNPs. Again, I'm not going to speculate on the significance of this. Let's move on to some methods. So primarily we're going to begin with methods that depend on changes in gel mobility, that is electrophoretic, let's say instead electrophoretic mobility, because there are other means of separating our molecules electrophoretically besides gels. In any case, there are physical attributes that we want to focus on that determine electrophoretic mobility. Principally length. Obviously the physical size of the molecule is going to affect its electrophoretic mobility. Base composition will affect mobility as well. Specifically, let's think about that, how base composition might affect single strand secondary structure. So of course, if we're talking about length, we're talking about size, but there are other factors that affect size as well, and that's of course secondary structure. Melting characteristics can also be utilized, that is to say the stability of a double-stranded segment of DNA. And of course, mismatches affect mobility changes as well, and this might be thought of, you can think of this both in terms of how melting characteristics are affected, but also in terms of how this affects size too. Size and also, let me say, shape. So all of these factors play a role in determining differences in electrophoretic mobility. We have a family of methods that we can talk about for identification and diagnosis of specific polymorphisms. That's again conformational polymorphism, referring back to all of the electrophoretic mobility determinants referenced above. Gradient electrophoresis methods that we can use to separate molecules of very subtly different shapes and sizes and characteristics, and then heteroduplex analysis, and we'll talk about all of these. But the bottom line is that what we're trying to do is introduce differences in mobility, in gel mobility. So we're trying to enhance those differences so that we can observe them as differences in mobility. Let's focus on single-strand conformational polymorphisms. And in this slide, I want to make it clear that I'm going to be using examples from the literature as a means of describing these techniques. So first, let me talk about what the method is, and then let me refer to the data that's shown on this slide. And obviously, this is a gel electrophoretic technique. In any case, cDNA is PCR amplified and heat denatured. So we're looking at single strands electrophoresed on a native gel. Mobility depends on length. Obviously, as we've said, that's going to be our principal separation method, but also on secondary structure formation. And I think it's obvious why secondary structure formation could affect the mobility of a gel, but if it's not, let's just consider what this kind of a structure looks like as compared to this kind of a structure, just in terms of of how it's going to migrate in the gel. So variants once identified, once found, are sequenced to determine the exact nature of the polymorphism. But first of all, we're just going to try to find them. So single-strand conformational polymorphisms combined with other methods such as restriction digests can enhance that sensitivity. That's obvious, I think, to imagine that the smaller the fragments that we're trying to understand, the more obvious the differences in their shape and size will be on the gel. Now, the data. So this is a paper from 2006, Immunogenetics, looking at porcine mannan-binding proteins. These are lectins, that is to say they're proteins that bind carbohydrates. Carbohydrate binding proteins. They seem to mediate, they mediate cell-cell interactions, but they're also responsible for some immune dysfunction. In particular, though, mannan-binding protein, so MBP, and I think in this case, most of it we were talking about some mannan-binding lectins, MBL1 and MBL2. It doesn't particularly matter, but the bottom line is that these molecules are going to be interacting with carbohydrates on the surfaces of invading bacteria. However, this immune dysfunctions that we see for mutants in MBL1 and 2 seem to suggest that there is some interaction with self carbohydrates as well that could be problematic. Let me just say that in terms of the upshot of the significance of these results, we don't know yet. This is an exploratory study, but what I'm trying to show you is that we can use single-stranded conformational polymorphism analysis to identify particular SNPs that seem to correlate with immune dysfunction. And in any particular lane here, we can see that there are here's a particular example of a polymorphism that arises as the result of or is observed as a result of a difference in that electrophoretic mobility. We see some more here, and we're highlighting out a couple more here. So again, these are just single-stranded restriction enzyme fragmented pieces of DNA that should be homogeneous, but in fact are not. And here again, so I'll just point that out again. So as a reference, you can compare the slide lanes 2 and 3, and you see that lane 2 shows two clearly separate species, and then we can sequence these and identify what the exact mutation is. And once again, it doesn't actually matter in the context of our discussion today. We don't care about mannan-binding proteins in pigs and the immune disorders of pigs. However, the point being is that we can identify these SNPs easily via this kind of technology. Okay, gradient electrophoresis. So there's a couple of different ways to introduce, again, greater sensitivity. So enhance sensitivity to size and shape changes, and again, those size and shape changes are conferred through changes in the sequence. We can use temperature, or we can use some kind of chemical gradient. But I think in this case, this specific example from molecular immunology, we're talking about temperature gradients. Again, amplification of the cDNA, and then we electrophorese the products along a linear gradient of temperature. And of course, the mobility of any particular species is going to vary depending on the temperature. So what we see is multiple bands, once again, that migrate with slightly different melting temperatures or melting profiles. And once again, I'm not going to interpret this data for you very much, but simply to show that we see a variety of differences and different species that emerge when we separate these according to temperature on the gel. In this case, we're talking about L-ficalin, so another carbohydrate binding protein similar to the mannan binding lectins that I discussed on the previous slide, also implicated in innate immune disorders. And I guess the only elaboration that I'll add here is that this autoimmunity that we're observing probably arises as the result of changes in what target molecules or target cell surfaces are bound by these lectins. In any case, SNPs in L-ficolin were identified in this paper by DGGE, and what we see is, we'll just take an example of this. If we weren't separating this molecule by gradient electrophoresis, temperature gradient electrophoresis, then what we would see is one species, and instead we see four. So suggesting to us that we have a variety of SNPs that we have identified by this method. Okay, one more example here of identification of new SNPs. Let's think about heteroduplex mobility. So heteroduplex, referring here to the fact that the duplex might be composed of sequences that are not perfectly complementary, and therefore will migrate with rates that reflect that difference. So again, that subtle change introduced by a bit of non-complementarity is going to result in a difference in the migration. So here's a completely complementary strand migrating here. Here's a strand that is heterogeneous. So there's both complexes here, and we see that here represented as two peaks. But again, it's a matter of amplification. So hidden behind here is a technique that we're obviously not going to talk about in this class, but just PCR, and then we denature that, re-annealing it with a wild type or some standard. That standard is going to define whether or not there's an SNP present. So and again, the heteroduplex now, that is to say wild type versus SNP or mutant DNA, will have a different mobility that we can observe. In this case, this is an example of capillary electrophoresis, not gel electrophoresis. And again, it's just a specific example of some real data. TX happens to be an aberrant Ig kappa. It's expressed in a particular kind of tumor, nasopharyngeal tumors. And the cool thing about this study is that we identified, I say we, Rin et al. Identified a risk-associated SNP, that is to say an SNP that seems to correlate with this tumor using this heteroduplex analysis method. Okay. See, let's say it over again. So 11 minutes and 23 minutes. All right, in this method, here's another method based on the varying mobility of DNA. In this case, we're comparing the duplex DNA that has no mismatch or substitution present versus one in which there is a mismatch. And you might think that I'm drawing. Now let's turn to heteroduplex mobility. And in this case, what we're talking about again is a subtle change in the structure of the now duplex DNA, and how we can monitor that subtle change as a change in the mobility of the DNA in some medium. In this case, it happens to be HPLC. In the heteroduplex, what we're talking about now, of course, is the same idea, and through all of these, PCR is hidden in the background, and we're not going to discuss PCR in this course, but essentially we amplify some products and we denature it and re-anneal. And this is where the homo or heteroduplex can come in. We anneal, and it depends on our choice of what kind of target DNA to re-anneal with. If we have a strand that's perfectly complementary, then we will have no heteroduplex. But the idea here is that maybe we have, in the case of a single nucleotide polymorphism, a mismatch, and that difference is enough to introduce a difference in the mobility when we run those products out on HPLC. So here we see the wild type, and here we see two populations, one wild type, one heteroduplex, run out on this HPLC chromatogram. Now, again, just to give you the details of what the data are, it doesn't necessarily matter so much. We're not interested per se in aberrant IgK expressions, but the point is that this particular molecule, TX, is expressed in a particular tumor type, and we can use this method of identifying SNPs, new novel SNPs, to find a risk association. So ultimately, perhaps leading to the development of some diagnostic tool in which we can use to determine what kind of tumor type we're talking about in a particular cancer patient.